Welcome to The Explainer. Today, we're taking a guided, behind-the-scenes tour inside the absolute most important microscopic mass production factory in the known universe. We're talking about the ribosome. Look, if you want to understand how life operates at its most fundamental level, how illnesses actually take hold, or how modern medical miracles are engineered, you have to understand this molecular machine. So, let's get into it.
You know, when we look at standard cell diagrams, ribosomes usually just get drawn as these tiny, abstract textbook blobs. But in reality, they are so much more than that. A ribosome is this highly complex ribonucleoprotein complex, meaning it's built from ribosomal RNA and proteins. And uniquely, it doesn't even have a membrane surrounding it. Honestly, you should think of it less like a passive blob and more like an ultra-efficient 3D printer for proteins. It reads the instructions and physically manufactures the actual building blocks of life, layer by layer.
To really grasp the stakes here, think about it like this. Ribosomes are the molecular machines that translate genetic information into functional proteins, making them essential for virtually every aspect of cellular life. Without this factory, your genetic code, it's just a blueprint locked away in a vault. It doesn't actually do anything. Without the ribosome actively translating that information into proteins for your cell structure, metabolism, and signaling, life as we know it simply ceases to exist.
So, here's our tour map for today. We'll be hitting 5 stops. 1. Meet the molecular machine. 2. The protein assembly line. 3. Bacteria, humans, and antibiotics. 4. When ribosomes malfunction. And finally, 5. The future of biotechnology.
Okay, let's dive into this. Section 1. Meet the molecular machine. Understanding the subunits.
Every single ribosome is built from 2 distinct pieces, a small subunit and a large one. The small subunit's job is basically to bind and read the messenger RNA, the mRNA blueprint. Meanwhile, the large subunit is the heavy machinery. It catalyzes the peptide bonds, literally linking the amino acids together. And because this bond-forming activity is actually performed by the RNA rather than the proteins, the ribosome is classified as a ribozyme. There's also a pretty crucial split across domains of life here. Prokaryotes, like bacteria, run on a complete 70S ribosome, while eukaryotes, like us humans, have a slightly bulkier 80S ribosome.
Now, what's really interesting about this is the math. A 30S small subunit and a 50S large subunit combine to make a 70S complete ribosome. Right, 30 plus 50 definitely does not equal 70. So what's with the weird math? Well, that S stands for Svedberg units. It doesn't actually measure weight, it measures sedimentation behavior. Basically, it's how fast these components sink to the bottom of a tube in a centrifuge. Because their shape and surface area physically change when the 2 pieces lock together, they sink at a rate of 70S, not 80S. The biological math absolutely defies simple arithmetic.
In our eukaryotic cells, these factories set up shop in 2 main locations, and their physical location dictates exactly where their manufactured product gets shipped. Free ribosomes just float around in the cytoplasm, churning out proteins that are going to be used locally, right there in the cytoplasm, the nucleus, or the mitochondria. But the ribosomes attached to the rough endoplasmic reticulum, the RER, they're manufacturing proteins destined for export. Those proteins are going to be secreted outside the cell, embedded in cell membranes, or shipped off to lysosomes.
All right, moving right along to Section 2, the protein assembly line, translating the blueprint.
This right here is the ultimate flow of information. DNA to mRNA, and finally, mRNA to protein. The assembly line itself runs in 3 super clear steps. First up is initiation. The small subunit binds to the mRNA, an initiator tRNA recognizes the exact start codon, which is almost always AUG, coding for methionine, and then the large subunit clamps down on top, forming the fully functional ribosome. Step 2 is elongation. The ribosome literally chugs along the mRNA strand, reading each codon and linking amino acids together into a growing folded chain. Finally, we have termination. The ribosome hits a stop signal, UAA, UAG, or UGA. There's no tRNA that recognizes these. Instead, release factors swoop in, bind, and free the completed protein to go out and do its job.
Now, to effortlessly remember how raw materials actually move through the factory during that elongation step, you really just need this simple memory trick for the 3 binding sites inside the ribosome, A, P, and E. A is for arrival. This is where the incoming tRNA carrying the brand new amino acid first arrives. P is for peptide. This site holds the tRNA that's currently anchoring the growing peptide chain. And E is for exit. This is where the empty, depleted tRNA leaves the factory. Arrival, peptide, exit. Raw material comes in, gets added to the chain, and the empty transport vehicle is ejected. Simple, right?
But biology is all about efficiency. The cell doesn't just send one single mRNA blueprint to one single ribosome. No way. It forms what we call polysomes or polyribosomes. This is where multiple ribosomes attach to a single mRNA molecule at the exact same time, moving along it like a long caravan. This clever trick allows the cell to rapidly pump out massive quantities of the exact same protein in a fraction of the time. It's literally the ultimate sign of manufacturing efficiency.
Which brings us to Section 3, Bacteria, Humans, and Antibiotics, exploiting the differences.
So why does knowing the structural difference between a 70S bacterial ribosome and an 80S human ribosome actually matter in the real world? Well, it matters because it's exactly how we fight off deadly infections. Familiar antibiotics are precision-engineered to intentionally jam the gears of bacterial factories. Drugs like tetracyclines and aminoglycosides bind straight to their small 30S subunit, while macrolides and chloramphenicol target their large 50S subunit. They physically block those A, P, or E sites we talked about, stopping bacterial protein synthesis dead in its tracks. So the crucial point here is selective toxicity. Because our human ribosomes are structurally different. Remember, we're rocking those 80S ribosomes made of 40S and 60S subunits. These antibiotics just don't fit into our machinery. That means we can safely take these drugs to completely destroy the bacterial factories and cure the infection without touching or harming our own cellular factories. It's incredibly elegant.
Let's pivot slightly to Section 4, when ribosomes malfunction, ribosomopathies, and disease.
What actually happens when our own factories break down or get hijacked? Well, when human ribosome biogenesis, which mostly happens in the nucleolus of our cells, goes wrong, it leads to a class of really severe systemic diseases known as ribosomopathies. We're talking about conditions like Diamond-Blackfan anemia and dyskeratosis congenita. Plus, if ribosomes lose their quality control, they can start pumping out incomplete or defective proteins. And that buildup causes intense cellular stress. On the flip side, a hyperactive ribosome isn't a good thing either. Changes in ribosome biogenesis are heavily linked to cancer development. It makes sense, right? Rapidly dividing cancer cells have massive, insatiable demands for protein synthesis. They absolutely need those factories running in overdrive just to spread.
Let's move to the final frontier and see how this builds into Section 5, the future of biotechnology, engineering the factory.
Because, we aren't just observing these molecular factories anymore, we are actively manipulating them. So how exactly are we engineering ribosomes for the future? It's a fascinating question.
It turns out, a ton of our modern medical miracles rely completely on hijacking this biological 3D printer. Through recombinant protein production, we insert engineered genes into bacterial or eukaryotic cells, and their ribosomes dutifully translate our instructions into life-saving therapeutics. The insulin in a diabetic's pen, growth hormones, and groundbreaking vaccines, all produced exactly this way. And in the realm of synthetic biology, scientists are even engineering these translation systems to incorporate completely non-natural amino acids, creating entirely novel proteins that nature frankly never intended.
But to pull all of this off, researchers need to see exactly what the ribosomes are doing in real time. Enter ribosome profiling, or Riboseq. This is an incredibly powerful molecular biology technique that essentially takes a live snapshot of which mRNA strands are actively being translated by ribosomes at any given millisecond. By monitoring that translation efficiency, scientists are bridging the massive gap between static genomics and dynamic protein synthesis. They are literally uncovering entirely new, previously unknown translated regions of our genetic code.
This leaves us standing on the absolute edge of an incredible biological frontier. From leveraging AI to predict complex ribosome structures to developing next-generation drugs against antibiotic-resistant superbugs, the potential here is pretty much limitless. Just think about it. If we can engineer these factories to print completely synthetic, non-natural proteins, what entirely new forms of synthetic cells and personalized cancer therapies are we going to build next? The tiny molecular factory that built every living thing around us is now in our hands. Thank you so much for joining me on this explainer. I really hope this leaves you wanting to explore even deeper into the microscopic machinery of life.